adjusting according to your height.
Yeah.
Should I use this one?
Maybe
Yes.
we will.
Okay. This is a pointer.
Yes, the top button. Do you need it? I can
Yeah, I
turn
can
I
it.
think it's better.
Then I can walk around a little bit.
Yeah, but then it's like an earpiece. You have to put it around your ear. I could have a
Yeah.
normal one.
No, but that's okay. I mean, look, you put it there behind your ears like this.
Yes.
Yeah, that's okay.
So shall
And
I turn it on?
Yes.
Yeah, because then if we turn back, if we turn away from here, then they don't hear. So this is better if it's well.
Yeah, sure. So
Okay.
I turn it on.
So shall we put it on now already?
Yeah.
Well, if you want.
Okay. Okay, just put down.
I don't know if it's better to put this in your pocket or
Ja,
keep it on the table.
ein bisschen.
Ich weiß nicht, wie es
Nee.
ganz funktioniert.
How do we do it? This way around.
Yeah, that's better.
Yeah,
Okay,
this is-
that's
yeah,
better.
that's better.
Okay.
Okay, so yeah, but it's not close enough. How
Is it all right?
It should work, I guess.
All right. Let's
no,
try it.
it's not.
If it doesn't work, leave it, I think, then I take this one in. Yeah.
Yeah, I think
Give me something into the end. That's just better. Okay.
It is working.
Is it?
This one was working, yeah.
yeah, but it's too small.
Yeah.
It doesn't. It's sort of. It is cheap. So I don't know.
We'll just
Hang
give
on,
you a
hang on. All this technique, we can. This can move.
Don't break it.
No, but this can move.
Test, test.
This one
Quite.
here. If you can help me, this moves.
No, I can take it. It's fine. But just make signs. Sometimes I have it a little lower.
Yeah, but then look at us, and we'll.
Okay. Okay.
Okay, so let's turn it on.
Let's do it. Okay. All right.
And this is the one to
Yeah.
and more industry.
Da-da-da.
Computer. And where do I move it forward and backwards?
Here we go.
Can we go back? Okay.
Okay, so here we go. We will invite our host and we will begin the competition.
You can check it if it's okay.
So hello and good morning everyone, and welcome to the second day of the Coins 2018. Please applaud. As we have a lot of people today, and today we'll be glad to invite you to our company fair, during which you'll have the opportunity to explore new career possibilities in science labs and companies throughout the country, and as well as, I'm sure, some very wonderful presentations. But before we begin, I'd like to add a quick reminder about the surveys that you should have found on your desks. We would be very thankful if you fill them in and throw them into this box at any time throughout the conference. And now, thank you for that. And now, without further delay, I would like to invite on stage a very esteemed guest indeed. Please welcome Harald zur Hausen. Please welcome.
Well, Lava Sristi, I hope I pronounced it correctly. Good morning, everyone. It's a great pleasure for me to be here, and thank you for the invitation to come here. Is it loud enough? I hope it's better now. You can understand me. All right, what I'm going to do today is to tell you, first of all, a little review of some work which we conducted, in part at least, for a very long period of time. For myself, almost for something like 50, close to 60 years. And we'll present some recent approaches to the question which is interesting us since a very long period of time, namely, to which extent do infections contribute to human cancer? That will be a specific question. And we just a little bit of an historical review. Can we dim the lights a little bit? Now, we do know today that approximately 21% of the global cancer incidence is linked to infections, to infectious events. These are in part viruses, to a relatively large part. That's a little bit too dark. And we start to sleep all. But viruses, but also bacteria, and in addition, some parasitic infections. Well, viruses, you have some members here of this group indicated. The herpes viruses, in particular Epstein-Barr virus, the human herpes virus type 8 in addition, papilloma viruses. Our group was engaged for quite a long period, for several decades, specifically in the study of papilloma viruses. And polyoma viruses, another group which received more interest in recent years because some human representatives have been found, specifically the so-called Merkel cell polyoma virus, which causes a rather nasty form of carcinoma, and hepatitis B virus. These are all DNA viruses. But besides those, we know a number of other agents, namely specifically RNA-containing viruses. And some of them are listed here in the flavivirus family, the hepatitis C virus, an important carcinogen, particularly also in Europe, but also in the retrovirus family, in particular the human T lymphotropic retroviruses causing a leukemia, in particular in coastal regions of Japan, the HIV, the human immunodeficiency viruses, which suppress the immune system and activate or lead to activation of other agents under those conditions which lead to cancer. And in addition, bacteria like Helicobacter pylori, an important agent concerning gastric cancer, and parasitic infections, here specifically the liver flukes, which cause specific types of liver cancer in gallbladder cancer and cholangiocarcinomas, particularly in Taiwan and southern China, and the schistosoma infections, which are important for bladder cancer, specifically in Egypt, in Africa. Now, a couple of different mechanisms by which these agents contribute to cancer development, and they are listed here. One of the initially recognized and important mechanism is the introduction of specific viral genes, viral oncogenes, into host cell DNA, frequently as integrated DNA into host cell genomes. And we do know quite a number of agents like papilloma viruses, Epstein-Barr virus, human herpes virus type 8, where the latter two usually do not integrate but are linked close to chromosomal DNA, or mainly linked to chromosomal DNA. And the Merkel polyoma virus, which was a very peculiar mechanism in its induction of cancers. Now, besides this, I mentioned already HIV, the human immunodeficiency viruses, which induce immunosuppression and activate other tumor viruses, specifically Epstein-Barr virus, but also a few other human herpes virus type 8 in particular, and a couple of other agents. The other interesting mechanism is really a relatively novel one. That's namely the induction of chronic inflammatory events, the induction of oxygen and nitrogen radicals, and this is particularly true for hepatitis C viruses, to a limited degree probably also to hepatitis B virus, to Helicobacter pylori, and to parasitic infections. And some of them even act apparently, or could act, by prevention of apoptosis, a self-programmed cell death, which is a mechanism which has not been really directly linked to human carcinogenesis for certain types of human papillomavirus types, and also the induction of chromosomal instability, which is a feature by many of these agents, but not only by oncogenic viruses, but also by non-oncogenic agents, like measles virus, for instance, and a few others, which has never been shown really, convincingly shown, to be linked to human cancer as well. So basically we can differentiate between those agents which act as direct carcinogens because they induce, they have to persist in the tumor cells with specific gene products, and the gene products are directly linked to cancer development, and indirect carcinogenesis, where something has been modified due to the persistence of the virus, mainly by interaction with oxygen radicals in inflammatory events. Now, we understand the pathogenesis of some of these virus-induced cancers reasonably well. For instance, for human papillomavirus, schematically outlined here, that an initially infected clone of cells, of basal layer cells in the epidermis or mucosa, develops a mutation which permits an escape from the immunological control of these types of cells. And subsequently, this clone proliferates more rapidly, and subsequently, additional mutations arise in a progressive way, which permit, first of all, an escape from an existing, I cannot go into the details right now, intracellular control of cell proliferation, but also from something which is an extracellular control, also of transcriptional activity of these agents, until finally cancer arises. And this process, in case of cervical cancer, takes about 15 to 25 years, from there to here, from the infection to the emergence of cancer. And that is one example only. We do know others where the latency period is much longer. For instance, here, as you can see it, for the hepatitis B and C viruses, for parasitic infections, also for Helicobacter pylori, the latency periods maybe end up to 30 to 60 or even up to 70 years after infection. Enormously long periods of time, which made it very difficult in the past to elucidate the original cause of those types of cancers. And it's now coming up that some of these have very long latencies indeed. The interesting part is here. We do know one single human malignancy at this moment, one single malignancy, the so-called X-chromosome-linked lymphoproliferative disorder, which has been linked to Epstein-Barr virus infection, where the existence of a chromosomal modification in the X chromosome prevents an effective T-cell response against these types of infections. And under those circumstances, the infections which acquire this genetically inherited this type of modification already from their mothers usually. Those patients suffer upon Epstein-Barr virus infection subsequently from an acute lymphatic disease, which in most cases led to death of the infected individuals. But this was a clue, really. What we learned from here was really that there exists no single infection leading to cancer which does not need in addition some additional modifications of specific host cell genes. Modifications either by translocations, by mutations, by epigenetic modifications. They're essential in specific parts of the genome prior to the development of a malignancy. And the length of the latency period here, in comparison to here, for instance, it gives you some relationship to the number of genetic modifications which have to take place in the very same cell. Now, this just as a kind of introductory remark. We do see right now globally an increase in the incidence of cancer, as it's shown here for males and females, and at the same time a somewhat slow reduction of the mortality from cancer, which is due to an improved treatment of cancer patients. The mortality is going down, the incidence is going up. This tells you instantly, if you really wish to do something against cancer, against the occurrence of cancer, you have to do something here to reduce the incidence of cancer. And the only way of doing this would be really, in case of infectious events, the prevention of those types of infection. And we do have ample examples for this, particularly for hepatitis B virus infections and also for human papillomavirus infection, where vaccination is available which prevents specific types of cancers. And we hope, of course, that many more will occur in the future. Now, in spite of a large number of cancers which have been totally sequenced in the past, well, 1-2 decades, really, since 2008, when the Merkel polyomavirus was discovered, there exist no consistent reports which pointed to novel infectious agents in human tumors. And this, of course, has one clear-cut consequence. Either we know by now the vast majority of infections which have been linked to human cancer, or, and this is the alternative, the involvement of infectious agents does not follow the characteristic pattern of well-established oncogenic pathogens. And that was our basic hypothesis when we continued to work after having worked for a long period of time on papillomaviruses in human cancer, to look into other types of cancer, whether there's any reason for this type of suspicion. Now, the basic background from these came from the following considerations. We do know in humans a number of infections, like the so-called BK and JC polyomaviruses, which are present in 60 or 70% of all of us sitting here in this room right now for any time, and for lifetime. Usually without doing anything, except under conditions of severe immunosuppression, they may have some consequences, which could be quite serious, but which I'm not going to discuss here today. The human adenoviruses, type 12, 18, and 31, and other agents, which are frequently present in human populations, never been shown to cause cancer in humans, and in humans where they can replicate. But if those agents are transmitted to species in which they cannot replicate, for instance here, rodents, hamsters, and owl monkeys, they're suddenly tumorigenic. So under conditions of a non-replication, when not all viral functions are being expressed in those cells, they are suddenly cancerogenic. And that led to the suspicion, is it possible that those animals with which we live in our close neighborhood, that they also acquired some kinds of infections to which they adapted during replication in those agents, in those animals, to which they adapted in the past for persistence without any side effects in pathogenicity, particularly in oncogenicity. But if they are transmitted to a species in which they are not replicating, where they become replication incompetent but are still infectious, whether under those conditions they could cause cancer. That was the basic idea. In fact, I published it in the year 2001, and in the Lancet there were 5 quotations of this publication up to today, and I found out that most of the citations came from myself in subsequent papers, so it didn't find any kind of interest at all. Now, this is basically the same which I said before. These are some of the starting points of our consideration. Let me skip this because it says the same what I said before. The main candidate for our considerations was clearly the look into the epidemiology of certain types of human cancer, whether we could see any kind of hints which could link those types of cancers to a potential role of infections. And human cancer of the colon was really the prime candidate in our studies. Why? Because it has been linked since a very long period of time to red meat consumption. A large number of reports describe a high incidence of this type of cancer. about 20 to 30% higher increase in those persons who consume red or processed red meat, air-dried meat, smoked meat, and so on. Countries with the highest rate of red meat consumption, they are listed here on the slide, commonly reveal a high rate of colon cancer. That's quite consistent with very few exceptions to which I come in a moment. Since 1978, it was quite clear, it seemed to be quite clear why this is the case. Because if you roast, barbecue, or broil red meat or meat, you get a number of chemical carcinogens being formed in the cooking or boiling process. In particular, aromatic hydrocarbons, nitroso compounds, and a couple of others which are listed here. And they are, if you feed them to rodents, to mice, usually at 10,000-fold higher concentrations than they're found in red meat preparations after roasting it, they turn out to be carcinogenic, not only for the intestines, but also for other organs in those animals. And that seemed to explain the story quite conveniently, that red meat consumption creates a higher risk. The problem of the story was really, and that was noted already by Sugimura, who discovered initially in Tokyo these types of carcinogens, that if you roast, barbecue, or grill chicken or poultry in general, or fish, and you follow up populations which mainly live or consume fish, for instance, or poultry, they don't have an increased rate of colon cancer at all. Some of them have a very low rate of colon cancer, quite in contrast to the other populations. And so we became very much interested in the epidemiology of colon cancer. And it's interesting in the geographic epidemiology. There are two striking features already from the beginning. One of them was an interesting one, India here, which has the lowest rate of colon cancer. Now India, you all know, one kind of meat is not being consumed, almost not at all consumed in India, that's beef, due to religious customs. And the other interesting country was even more interesting in a sense, because that's Mongolia. Mongolia has approximately the second lowest rate of colon cancer and has one of the highest rates of red meat consumption in the world. And that didn't fit at all into the whole story. Now, of course, we were interested which kind of meat is being consumed there in comparison to the other European or Asian countries. And it turned out, this story turned out to be also quite interesting, because they eat a lot of so-called beef, but this beef is a different species of beef, yaks in particular, a very different type of beef, and also a kind, what they call the original Mongolian cattle, which is was genetically different from the European type of cattle, but which was later, since the 1950s, crossbred extensively with zebu cattle, the Brahman type of cattle from India and Southeast Asia, and are now mainly really zebu-derived types of cattle. And the zebus are also genetically different, both indicus from the European and Eurasian type of cattle. So to make a long story short, wherever the European type of, Eurasian I should better say, type of cattle is being kept, you have a high rate of colon cancer. It seems that it's quite restricted to this region. In China, they started in the past decades to eat more beef, but also different types of beef. But the main source of meat is clearly pork in China. 80% of the meat consumption appears to be pork. Bolivia is another interesting country, because in Bolivia, you have almost exclusively zebu-derived types of cattle, which are, again, it's a different breed in comparison to the European cattle. To make a long story short, we suspected there's something specific in the Eurasian type of cattle, or good dairy cattle, which, of course, the product is not only the production of meat, but also the production of milk, which at least was suspicious to us. So we started to look into the question, are there, besides colon cancer, other cancers which have been linked either to meat or dairy product consumption? And the list is quite long. And it's also long for other types of diseases, not only for cancers. The consistency of observation is high for colon cancer. It's moderately high for breast cancer, even for lung cancer, interestingly. Lung cancer in particular, since a group in Sweden demonstrated that in lactose-intolerant people, which they compared to their siblings and parents, they have a rather drastically reduced risk for breast cancer, for instance, and also for lung cancer. Prostate cancer, milk consumption has been repeatedly linked, and a couple of others where the evidence is not so clear-cut. I wrote down here a low relationship to these types of cancers. Now, so our analysis of the global epidemiological pattern of colon as well as of breast cancer suggested that the consumption of meat and dairy products of the so-called aurochs-derived farm cattle are prime candidates for the risk of these types of cancers. And we published this repeatedly and collected some additional evidence from other pieces of observations. Now, in 1914, our group published a number of isolations of specific circular single-stranded DNA genomes from serum and dairy products of Orisian cattle. Here I should say that Esther Michelle Devilliers, my wife, she's here at the same meeting, did a lot of previous work on TT viruses. These are agents which are also small single-stranded DNA viruses which virtually infect 100% of the human population. And we used the techniques which she developed for the TT virus isolation to look into the bovine serum preparations and also into bovine dairy product preparations to isolate those agents. There are a couple of publications which appeared which described the isolations of those types of genomes which we did before. Basically, in the early studies, and you will hear from Esther Michelle a little bit later more intensively about these agents, which we started to group into 4 distinct groups. We called them BMMF, bovine milk and meat factors, or bovine meat and milk factors. Group 1, 2, 3, and 4, whereas group 3 is a typical virus isolation, which concerns the circovirus group. The other 2 are structurally different, and amazingly enough, they contain sequences closely related to bacterial plasmids. And that, of course, instantly created the suspicion that these are bacterial contaminants and have nothing to do with the diseases yet. So we used those types of genomes and transfected them into human cells. And amazingly enough, it turned out that all of those which we transfected, first of all, were able to be transcribed in human cells. These small genomes with about 1,300 or 1,200 up to 3,000 nucleotides long. They were genetically active in human cells, and they are also transcribed. They are also translated into a specific protein in human cells as well. We know today from work of Konstantina, who worked in our group, the name is misspelled here, Falida is her last name, that indeed in some of these lines, in specific lines, there's even an active replication taking place after continuous keeping these plasmid-like structures in these types of cells, and they replicate within this period of time quite extensively and form even some types of, in the later stages, some types of defective genomes which usually cover a specific part of the genome. Now, this, of course, raises an interesting question. Do we acquire infections from bovine milk consumption? Well, the epidemiological data suggests, and I will come to them in a moment, that events early in life could play a significant role for the risk of some of these cancers and also for some other chronic diseases. And it has been known since quite a long time that the prolonged duration of breastfeeding is an important protective factor. Let me come to this in a little bit more detail right now. Protective effect of prolonged breastfeeding, more than 6 months usually, has an effect on the following chronic diseases: a moderate effect, clearly an acute lymphoblastic leukemia. In those breastfed children, lymphoblastic leukemia occurs less frequently. Hodgkin's disease is clearly affected by long-time breastfeeding. Multiple sclerosis, which I will not cover today, although it's another interesting topic, there's also reasonable evidence that prolonged breastfeeding protects against multiple sclerosis to some degree. In other diseases, the evidence is much less convincing, particularly in diseases which occur later in life, premenopausal breast cancer, for instance, because it's usually very difficult for those individuals who acquire these cancers to remember how long and to which extent they've been breastfed in the past. But even diabetes belongs into this category. Now, there are some very interesting observations which have been made in the past about the reasons for the protective effect of breastfeeding. And interestingly, during the breastfeeding period, let's say here for the first 6 months after delivery, there are a couple of sugars present in human milk, the 2' and the 3' fucosyl lactose, and also a tetraose, which obviously exerts this protective effect. And this is partially understood by now, because they become incorporated into glycoproteins and gangliosides, which act as receptors for some infections. During this period of time, the babies are not susceptible, or at least not very susceptible at all, against rotavirus, norovirus infection, and a couple of others, even specific influenza virus infections or HIV in some instances has been described as a protection exists during this period of time. And only during the weaning period, when you subsequently start to add dairy products to feeding the babies, then something else is happening. And this something else is quite important and interesting to know, because at this stage, we take something up which is not present in human milk, but which is present in cow milk, present in many other species. It's present in serum and in meat preparations for many other species except for chicken. And this is a very specific type of neuraminic acid, which has been described, and that's the so-called N-glycolyl neuraminic acid, the Nu5JC as it's frequently being called, which originates from a conversion of this N-acetyl neuraminic acid into this type of enzyme, of neuraminic acid, by a specific kind of enzyme which is missing, which is not being produced in humans. We have a deletion in one of the exons of this enzyme which does not permit this conversion, but it's present in almost all, not in all, but in almost all animal sera and dairy products as well. So this type of sugar, or neuraminic acid, sialinic acid, is taken up. It's also incorporated into glycoproteins and gangliosides, and it forms novel receptors for infections. Suddenly, these cells become susceptible for infections against, they have been not sensitive, like human polyomavirus type 9, several parvoviruses, influenza A and parainfluenza viruses, against which they were not infectious, and for which they were not permissive prior to this period when they were still breastfed in this time. So humans develop an immune response against these novel receptors, this novel final determinants of the receptor, and this is called xenoimmunization, has been studied particularly by a group in Seattle by Waki and his colleagues, and they called it xenoimmunization, which occurs. Initially they suspected that this was a reason for the susceptibility of red meat consumption and cancer development. But we, of course, instantly thought that it might be due to a specific uptake of something which leads to cancer, of an infectious agent leading to cancer in these individuals. And there's some reason to assume this. First of all, this is again the structure of these types of sugars which are present in the human milk, which obviously form the new elements on those receptors and enabling them to be receptors for novel types of infectious events. There's one very interesting observation which came up during the past decades already, that multiple pregnancies have a protective effect against some specific types of cancers. First of all, it was described for human breast cancer. There's an extremely large study done in the United States where they studied more than 340,000 deliveries. The mothers had deliveries on the number of deliveries which they had and the risk for breast cancer occurring subsequently. And what you can derive from this table is that there's about 40% reduced risk for breast cancer in women who had about 4 or 5 deliveries. An interesting phenomenon which has been partially ascribed to some hormonal effects of the deliveries, but you could have a very easy alternative interpretation for those types of events which happen here. Namely, during the first years of life, schematically in the first months of life, schematically indicated here, you have these specific sugars being produced, causing a resistance against some kinds of infections. It was tempting to assume that in subsequent pregnancies, now there's a protective effect for the mothers as well of the individual pregnancies, in part due to the same production of sugars, first of all in the breast. We assumed that it would also happen in the blood of those mothers. In fact, as far as the breast is concerned, there clearly is ample evidence there's a large production of these sugars in the breast, apparently in the Golgi apparatus of the breast. In the blood, I didn't find a publication, but in the urine, these sugars appear too, as well, which obviously is an indication that they must be in the blood as well. So this raised the suspicion that the blocking of 5-new-GC, of these specific neuraminic acid carrying receptors, and that in these cases prevent some kind of infections which may be linked to the previously shown risk for the development of breast cancer at least. Interestingly, there exists also some evidence for a reduction of the risk in colon cancer, not so dramatically, but to some degree as well in colon cancer. So this is another scheme which indicates this, that we have during the weaning period human milk factors, sugars, which block specific receptors, and as shown here on the left side, and on the right-hand side of this picture, that the uptake of dairy products, bovine meat products, and the weaning period, the stop of the breastfeeding period, that under those circumstances, a xenoimmunization occurs, uptake of 5-new-GC, and its incorporation into glycoprotein gangliocytes is a consequence, resulting then in some specific type of infections which remain latent, persistent in the infected individuals, and cause very slow chronic inflammatory events over decades. That was the hypothesis which was derived from these. So since we isolated specific BMMF, bovine meat and milk factors, members from colon cancer biopsies recently, and in particular from peritumoral tissue, this directed our interest into a possible mechanism by which cancer of the colon might be initiated. And so we developed against a specific major protein of the BMMF1 group specifically, monoclonal antibodies, and started to stain tissues of colon cancer patients and of controls for the respective antigens by using 3 sets of monoclonal antibodies which were all directed against different epitopes of the same protein. And under those circumstances, as you can see it here, the interesting result was You see here the Leberkühnsche crypts, as they are called, in the colon. And you see here the mesenchymal tissue. We found only the staining against these antigens by using different sets of these monoclonal antibodies in the adjacent mesenchymal cells and not in the Leberkühnsche crypts, in the cells from which the cancer arises directly. This was interesting. It was confirmed in a number of additional studies, as you see it here, very regularly arranged around the Leberkühnsche crypts, as in this case. But occasionally there was a suspicious cell in the Leberkühnsche crypts, but we do not know whether it's an artifact or not. But basically they seem to be free of it. It's interesting that in some of these instances we had very big clusters of this material, and these are some others. I will go to them quickly. The suspicion arose that this is a very specific mechanism which may lead to this type of cancer, namely that in these, particularly after seeing that the same islands which are stained here, I'll show it to you in a moment, that the same islands are being also stained by CD68, an inflammatory marker for macrophages in this case, that are also stained by CD68 antibodies. And so these are obviously inflammatory lesions here, probably very slowly producing oxygen radicals under those circumstances, schematically outlined here in this slide, which apparently affect both types of tissues, those here, but also the Leberkühnsche, the adjacent cells of the Leberkühnsche crypts. There's one big difference between the mesenchymal cells here and the Leberkühnsche crypt, because the mesenchymal cells here, particularly in the lower and middle part, the distal and proximal part of the crypts, show a very high degree of DNA replication at any time in these cases. Whereas if you calculate it for the mesenchymal tissue beneath it, there's at least a 100-fold difference in replicative activity, stained here by the Ki68 monoclonal antibodies, which stain specifically proliferating cells under those circumstances. So the suspicion arose here that the inflammatory events taking place here over long periods of time in a very slow process produce oxygen radicals which affect specifically those cells or induce mutations in those cells which are mitotically active, whereas in those more resting types of cell culture, cells surrounding the Leberkühnsche crypts, it's not the case. This shows in another picture the replication of the DNA here in the tangential section of the Leberkühnsche crypt, and if you reach them at the lower level, at the most distal level, you see that virtually every cell is in a proliferative state at a specific phase, whereas in the subsequent upper levels, there's no replication taking place. They're gradually coming up, migrating upwards, and then stopping their mitotic activity. Here you can see it better what is happening under those circumstances. This is just a staining of a carcinoma lesion under the same types of condition. Now, here you have an example that's the same region, which is being found positive by the monoclonal antibodies. If you stain it for inflammatory markers, you see here there's a rather relatively strong inflammatory reaction at the same time. And this shows basically the same picture which you have seen a moment ago. So this led to a kind of a proposed model how colon cancer develops. That we have initially a rather specific trigger by one of these agents which we call BMMF1, quite unspecifically for the whole group, because we are not yet sure which is really, whether there is a very specific one or whether several agents which are linked to this type of effect. That this subsequently results as the primary target cells in the infection of the mesenchymal cells of the colon, a focused spreading of the infection. It's to a subsequent invasion of macrophages, a chronic inflammatory response, as shown here on the right-hand side, a slow induction of oxygen radicals, and also, as we know, of COX-2 under those circumstances, induction of random mutations in adjacent colon carcinoma, in adjacent colon cells, colon carcinoma target cells, random mutations occurring now in these types of colon cells, and only if specific hits occur in growth regulating genes of those types of cells after a prolonged period of time, we calculate 30 to 70 years, carcinomas will develop. Interestingly, what we anticipated from this type of speculation, that colon cancer patients should show high antibody titers against the respective antigen. So we tested controls and those colon carcinoma patients. I don't show you the data right now, but they're quite intriguing. The opposite happened. The colon carcinoma patients have lower antibody titers against the respective antigens than the controls. Controls were, in average, higher than the colon carcinoma patients. And this is suspicious of a situation quite reminiscent of hepatitis B virus infection, perinatal hepatitis B virus infection, where you get an immunotolerance induced under those circumstances, or partial immunotolerance induced, where only at higher age some antibodies appear against specific antigens of hepatitis B. So it stresses again the fact that the infection probably is happening in all these cases in a very early life phase, namely probably in the first year, most likely in the first, after the end of the breastfeeding period of the women. And this tells you another thing. Presently you see a large number of epidemiological studies which are being conducted in 20, 30, 40, and 50-year-old women for the risk of breast cancer or colon cancer in relation to the nutritional habits which they have. If this what I'm told you here right now is correct, then this is all nonsense. It will not provide any useful results, which, if you look into the literature, looks exactly like that. There are no really, really useful results readable out of those data. And because there's sometimes a slight elevation, sometimes a slight reduction of the risk, and which is being linked, and it led to a flood of papers, of epidemiological papers, which, if you don't include the very early life period in these types of studies, they will not provide for you with any useful results. So this is just another scheme of colon carcinogenesis. There seems to be a non-random event happening in the first year of life, uptake of BMMF, after and during the weaning period of bovine meat and dairy products, infection of mesenchymal cells of the colon here, local spreading of the infection, induction of inflammatory response, production of oxygen radicals, a random induction of mutations in adjacent proliferating cells, and cancer appearance after 30, 60, or even to 70 years, with no persistence of viral genome in cancer cells. In fact, we didn't find in any of the carcinoma lines, which we studied so far, of colon cancer, evidence for the presence of this DNA, which was initially quite disappointing for us. And the partial immunotolerance, which seems to exist there, would readily be explained by an early infection of the babies by these types of things. We suspect that there are a couple of other cancers where such kind of infections may play a significant role. You see them here on the right-hand side, but this requires further investigation. This is a whole group which is doing these types of studies. As Michelle, whom you will hear in a moment, will talk about the agents themselves in more detail than I did. And we have external collaborators, pathologists from Zurich, Achim Weber, and also Matthias Heichenwälder, an immunologist, virologist, also at the Cancer Center in Heidelberg, and a couple of devoted people, which are listed here, which are quite intensively engaged in these types of studies. And these are our external collaborators. they're all the cows from the University of Leipzig, from which we got 130 sera, and which really initiated this type of study on the cattle materials at all. So in summary, we believe that we can establish here a kind of a novel group of pathogens, which are neither viruses, typical viruses, nor typical bacteria, but these are bacterial products, plasmids from bacteria, which obviously adapted to independent replication in human cells. There exist, of course, in our history some examples of similar events, like our mitochondrial DNA probably originated from a similar mechanism. The plasmids in the plant plasmids, which are responsible for the green color of the plants, are also apparently initially acquired by this way. And it certainly is a novel area of research that opens, hopefully, a number of new approaches to study other diseases as well. Thank you for the invitation, and thank you for...
So thank you very much. And do we have any questions? I see one hand. Yes? Please wait for the microphone.
Here. Hello, Professor Hausenas. I remember it was year 1999, you know, you got this prestigious Brubacher Prize in Zurich, I guess, you know, and you gave also a talk there. And 10 years later, I think there was another symposium where one of the speakers, Michael Stratton, he actually presented the data where he showed that he sequenced many cancer genomes, and he showed that there are passenger mutations and the driver mutations. And then basically, if you have a cancer, so it's accumulation of the mutations, where the driver mutations actually they driving the cancer, but most of these driver mutations, they are in the tumor suppressor genes. So basically, you knocking tumor suppressor genes, and you cannot target the cancer because you have no basically target, because there is no gene there. So my question would be, where would you put the money, you know, in terms of this, you know, is it nutrition, is it targeted therapy, or it's immunotherapy basically, where it's now developed by Novartis, for example, these CAR T cells which can target a tumor, you know, directly by immune cells like T cells, you know, which basically would be not dependent on anything, but they can do it. So how would you actually, where the money would go in your situation, you think?
I wouldn't bet any money on anyone here in this case, because both of these theories do not contradict each other at all, because the difference is only a specific one. That is the one that we postulate a specific trigger of the inflammatory events, and the random events which subsequently, due to this trigger, are being induced in the adjacent tissue. And of course, if you hit there a specific driver gene under those circumstances, which is a rare event, otherwise we wouldn't have the long latency periods for breast cancer, then you may have these types of subsequent changes in these cells, but you may also have it by a continuous exposure to these mutagenic events, which occur over the decades subsequently, that occasionally you have this one or that one, a suppressor gene or driver gene, as you call it, or Stratton called it, hit under those circumstances. So basically I see no contradiction whatsoever, except for the fact that we postulate one earlier event, which triggers really, initially, those types of mutations. Otherwise, it's everything fine.
And maybe the second thing, now big companies like AbbVie, you know, and Gilead, you know, they developed new drugs, you know, against, for example, hepatitis C, you know, where basically it's very expensive, but still we can eradicate. So you think we could go further, you know, and look for some compounds, you know, which would, for example, kill the papilloma or other things, you know, quite early that we would not develop? You think that strategy would work in principle?
I think it could work, yes, it's clearly so. But keep in mind that, for instance, for colon cancer, we know already that two events which block inflammatory reactions, namely aspirin and COX-2 inhibitors, have an anti-colon cancer effect. Slight one in COX-2, and they have create heart attacks in some instances, so it's out of the market right now for those purposes. But aspirin is still continuously being used, and this blocks inflammatory events, and under those circumstances the risk of colon cancer is reduced. That has been statistically documented. So this fits extremely well into our scheme, which I pointed out here.
I see one more, yes?
I want to ask if it is not any specific role of lipid raft for the entry of this carcinogenic virus. Because I know, for example, HIV use lipid raft as entry platform in the cell. So it's like human papillomavirus of any other of this virus. Lipid raft has an importance. Well, do you think that avoiding the entrance of the...
Let me not be misunderstood. There are clearly some specific genes in colon cancer, for instance, APC genes and a couple of others, which are really important for the development of this type of cancer. But what I'm postulating here is that the mutations in those genes are triggered by an event which is taking place outside of directly of the cancer cells. It took place prior to the development of these cancer cells just by mutagenic radical exposures, which occurred in proliferating cells. And that occurs, probably accounts also for many other cancers as well.
Thank you. And I think we have time for one more question. And do I see any hands?
It's widely known that colon cancer tends to form metastasis to
Excuse me, I'm
liver.
sorry, I missed it.
Metastasis. Yeah, okay. It's widely known. So can these infectious agents have impact on these metastases or on cancer spreading into other tissues, such as...
Well, theoretically, yes, but probably, I think, when cancer arises, it's almost a kind of an autonomous process where additional mutations arise with each subsequent division. So I doubt it whether these agents still have an effect on the metastatic development, but I cannot exclude it either.
Thank you.
So thank you once more. Please don't run away, as Justina has some presents for you. And now we'll have another esteemed guest for our next keynote presentation. And a quick reminder for our school students that after the presentation, please kindly leave through this second floor to avoid logistical problems. And now, once again, please welcome Ethel Michel de Villiers. Please welcome.